So moving on to our next topic, this is called Mendelian inheritance. And Mendelian refers to a scientist from the 19th century, Gregor Mendel, who gets a lot of credit, really earned credit, for figuring out a lot of the rules for genetic inheritance. We talked previously about the laws of inheritance, the segregation of alleles, independent assortment. These are things that Gregor Mendel really helped figure out by doing a whole lot of controlled genetic crosses. So in many ways, he was the first person to really lay out genetics in the way we think of it today. Our introductory slide here is showing a illustration of some of these inheritance laws. To zoom in on a variety of corn or maize that has variety in the variation in the colors of the kernels. So a little botanical tidbit for you. Every kernel on a cob of corn is the result of an independent fertilization event. So the mother plant is the same, and she contributes the ovule or the egg for the corn kernel. And then independently, pollen grains arrive on the wind, and they would fertilize those kernels independently. So they might come from different fathers, or they might just have a different genetic combination from the fathers. You can actually have controlled crosses in corn, and this would be the result of some heterozygous parents. So at some frequency, we get these very dark purple kernels or almost black kernels. And at some frequency, we get these yellow kernels, and the proportion of yellow and black kernels, something you can actually predict with really good accuracy. So it's a mathematical version of genetics. All right, so there is our scientist, Gregor Mendel. He was a monk in the Czech Republic, and actually his life overlapped a whole bunch with the life of Charles Darwin. So Darwin gets credit on the evolution side of biology for discovering a whole lot of phenomena in nature, and Mendel gets credit on a different side. They didn't really interact. There wasn't a lot of internet usage back in the 1800s. But they were roughly aware of each other, and certainly their contributions taken together these days, we can understand evolution in terms of genetics and genetics in terms of evolution. And it's really rich biology we have these days. All right, so his study organism was pea plants, and he set up garden experiments where he would control the pollination of pea flowers. He had varieties of pea that were able to be crossed and develop these phenotypes that were easy to tell apart. So in some ways, he was sort of lucky to have worked on phenotypes that were easy to discern. You could also say that he probably had a lot of trial and error, and luck was not so much in it as perseverance and making sure that he did have something that would work out in his favor. So imagine for a second that you are a Czech monk in the 1860s trying to figure this out. You've got two kinds of pea plant, right? People have dwarf pea plants, and you can grow dwarf plants from your dwarf pea plant seeds. And you've got tall pea plants, and you can grow tall pea plants from your tall pea plant seeds. Wow, what's going on? What is the reason that the dwarf plants are always dwarf and the tall plants are always tall? So he had a question. Some questions worth asking. So as I mentioned, lots of variation in the garden pea plants. So your book highlights some of the different genes that were involved. Here are a couple as well. Pod color, yellow versus green, pod shape, inflated versus constricted. So these are helpful, what we call Mendelian phenotypes. So sort of ones that worked out in his favor as being very clean differences, sort of one or the other. So yellow pods or green pods, no intermediate phenotypes. And as we now know, and as he figured out, these are the result of single genes. So a single gene affects whether you've got these yellow pods or green pods. And the yellow pods are the result of having two recessive alleles. So a genotype that is homozygous recessive is one with two little g's, and that gets the yellow phenotype. A genotype with any number of big G's, two big G's, or a big G and a little g, either homozygous for big G or heterozygous Those give you green pea pods. And that figuring out these genotypes, that was really the result of Mendel's work, being able to use this notation to figure out what's going on. So alleles can cause these differences in phenotypes or appearance. So the appearance of an organism is its phenotype, and phenotypes are the result of allele combinations, as illustrated here. Some of those alleles are known as dominant alleles. A dominant allele, as long as there is one of those, at least one dominant allele, the dominant phenotype shows through. So at least one big G gets you green. At least one big I gets you the inflated pod shape. Whereas the recessive alleles, they have to be homozygous to get you the recessive phenotype. So little g has to be homozygous to show yellow. Little I has to be homozygous to show you constricted. So I think we covered homozygous and heterozygous pretty well. And another term that's going to come up is a little bit more kind of colloquial term, true breeding. So to breed true means that if you had a group of these pea plants, let's say you had the yellow pod pea plants, and you always took the seeds from the yellow pod pea plants and let those plants mate together and kept planting their kids' seeds over and over and again, you would keep having yellow pea pods. So the true breeding means that they've basically got only one kind of genetics. A true breeding version of the green pod would be ones that are always green. When you cross green with green, you always get green. And those basically are terms. This term true breeding is synonymous with homozygous. So true breeding means, especially for these dominant phenotypes, means that they don't have any of the recessive allele in there. And that's kind of the starting point for what Mendel used in his experiments. So this is the organism, the garden pea plant, Pisum sativum. And in lab, you guys will talk about the model organisms that get used in biology. This one isn't really used these days as a model organism, but perhaps you could think of it as the first model organism, right? First organism that genetics was really worked out in. And not maybe the easiest. I'm sure there are reasons it's not currently preferred as a model organism. One of the things you might have to worry about if you're working on garden peas is that plants, a lot of plants have hermaphroditic flowers or bisexual flowers. So a single pea flower produces male gametes and female gametes, and self-pollination is possible. So you want to make sure that your pollen from one flower doesn't fertilize the ovules on that same flower. So this first step here is a step called emasculation, cutting off the male parts. So if you catch them early enough in development, before the pollen is mature, you can cut the pollen, cut the stamens out, and the pollen won't be contributing to that flower. So this purple flower here has been emasculated, and then you can take pollen from the parent that you want to be the male parent in this cross, get it on your paintbrush, dab it onto the female receptive portion of the flower, and basically control who the male parent is and who the female parent is. So controlled genetic crosses, we're going to do this in the fruit fly lab. We're going to control who the males are and the females are. We're going to make sure that the females are only crossed with the males that we intend for them to breed with. And this is an important part of genetic study, of course. If you don't know who your parents are in the cross, it's really hard to draw conclusions and be rigorous about what you're going to get for your results. So controlled genetic crosses, as we learned more about genetics, of course, then people would start with known genotypes, and that's what we're going to do in our in the lab. But you also, of course, can start this kind of cross with unknown genotypes as you try to figure out what the genes are that are responsible for your phenotype. Okay, so some examples of what Mendel worked with. We've shown the tall and short pea plants, tall and dwarf. The slide previously showed you differences in pod shape. There are also differences in seed color, seed shape, seed coat color, and so on. So basically, by dissecting little bits of this plant all across its plant body, Mendel was getting familiar with variation that could be genetic. And turns out a lot of this was genetic, and he figured that out for us. So again, as we've seen before, these are useful because they're cleanly easy to tell apart. Even something like tall and short pea plants, there just aren't intermediate sized plants in the ones that he was working with. And so that's a feature that could be told apart between different genetic genotypes of the plant. If you're curious, I think it's kind of neat to update sometimes what we hear about Mendel. These alleles and the genes that we talk about, we usually just kind of have a stand-in for them. We talk about them as, big Y, little y. But those are genes that are part of a much bigger organism with many more genes that aren't necessarily easy to study. And this is just a nice, I think, illustration of the genetic control of something that Mendel studied and something that we understand a lot better. So the difference in tall versus dwarf is not just like big T, little t, but you could break it down into a little more detail. It turns out there's this gene that gets abbreviated LE for stem length. And you don't have to know the details here, but gibberellin 3 beta hydroxylase is something related to basically plants' hormone signaling about when it's a good time to grow. So signaling growth, there's a difference, there's a genetic difference, and that causes this tall and short variation in the pea plants. And we know a lot more about the reasons why this single gene should cause plants to be tall or short, way more than Mendel ever could figure out. Okay, so we talked about controlled genetic crosses, having control over who the parents are, and exact transfer of gametes from one parent to another. And I want to show you just an illustration of the process. This is a very typical way to start a genetic experiment. So whether you know your parent genotypes or not, this is a good way to get in there and figure out what's going on. So let's say we start with a tall plant and a dwarf plant like we did before. The first generation is called the parental or the P generation. And this involves crossing individuals that are true breeding for different traits. So a lot of times this kind of cross would be done. If you don't know the reason why an individual would be tall and dwarf, you take your collection of dwarf individuals and your collection of tall individuals and cross them together and see what happens, right? That's sort of the genetic version of getting started. Cross them together, see what happens. So the P generation involves crossing two genetically different individuals, and then their offspring produce the what we call the F1 generation. It's F for filial. Filial is from the Latin for son or daughter, and F1, the first generation of kids from the parent. And it turns out that the F1 generation in this cross, if you cross tall pea plants by dwarf pea plants, then the F1 generation is entirely tall. So this genetic information for being dwarf appears to be lost, right? So this was a new finding for Gregor Mendel. Disappears, completely disappears, and all we see is the tall phenotype. Of course, what you and I know, of course, now is that these F1 individuals, they have a combination of a dominant allele from the tall parent and a recessive allele from the dwarf parent. And that dominant allele is sort of hiding the recessive allele and covering it. So these individuals, even though they look like the tall plants of the parental generation, are genetically different. They would be heterozygous and showing the tall dominant phenotype. So finding out more about these individuals, you take a cross of F1 by F1. So if you cross two individuals of the F1 generation, then that gives you what we call the F2 generation. And in a cross of two F1 individuals in tall and dwarf pea plants, it turns out that you get approximately a ratio of 3 tall pea plants to 1 dwarf pea plant. And this is a nice mathematical ratio. So when you do this cross, you could do it over and over again. Different starting parents that have the same genotype, you can do this repeatedly and always get this basically 3 to 1, 3/4, 1/4, 3/4 tall, 1/4 dwarf, 75% tall, 25% dwarf. And these would be differences in the phenotypes. And we'll see that when we look at genotypes, they're not necessarily all the same genotype for these tall pea plants. So as we've seen, it's possible to be heterozygous or homozygous dominant and have the same phenotype. So these tall pea plants, we know that they have at least one big T allele. We don't know if they're homozygous, big T, or heterozygous, big T, little t. So how do we figure that out? Well, there's one way to do it, which is to do a controlled genetic cross with the unknown individual crossed by a dwarf individual. So the tall pea plants could be heterozygous or homozygous, and if we cross them by this dwarf pea plant individual, that would tell us if there's any little t as part of the genotype. So crossing by dwarf here, might have to illustrate this for you a little bit later.